Hi, I'm Zoe Waller and I'm a Professor in Drug Discovery at UCL School of Pharmacy. This video is to explain our recent research paper, hopefully in a simple way. It's just been published in a journal called Nucleic Acids Research. The links to the paper are included on the YouTube webpage. This paper is called i-Motif stability, not G-quadruplex stability, modulates insulin expression. And it's published in a journal called Nucleic Acids Research. This journal publishes the results of leading-edge research into physical, chemical, biochemical and biological aspects of nucleic acids. So what are nucleic acids? Nucleic acids are essential biological molecules that store and transfer genetic information. For example, here we have some DNA. DNA is made up of four building blocks known as A, T, C and G. DNA is packaged in the form of chromosomes, which are found in the nucleus of cells, which make up the different tissues in our bodies. Let's go back to our DNA, made up of the building blocks A, T, C and G. DNA is known to be shaped like a twisted ladder. We call this a double helix. When the DNA contains a lot of the C and G building blocks, instead of being a double helix, we know that it can form different shapes, and we call these i-Motifs and G-quadruplexes. Previous work in our group showed that these special DNA structures could affect gene expression in the insulin gene. If the special shapes of the DNA were present, this turned the gene on. If these special DNA shapes were not there, the gene was turned off. This led us to ask the question, which is more important, the G-quadruplex or the i-Motif? What is the balance between these different special structures that DNA form? We performed experiments in cells using a system called a reporter gene. Our model was based on the human insulin gene. This meant that we could mutate the sequences that fold into the special DNA shapes. We found that for the gene to work, it needed both the G-quadruplex and the i-Motif structures. So we think both of these structures are important. We think this is because they work together to make the gene function. We then looked at the strength of each of these structures. This means how much effort we need to unfold them, and then looked at how that related to and correlated with how much of the gene they made. When we used stronger G-quadruplexes, this did not change how much gene expression we saw. However, stronger i-Motifs gave much more gene expression. In fact, there was a very strong positive relationship between the strength of the i-Motif and how much gene was expressed in our experiments. This means that the i-Motif controls gene expression in our experiments. The stronger it is, the more gene you get, a bit like a volume dial. We still consider the G-quadruplex obviously to be important. We think of this more like a switch. This work gives us a deeper understanding of how these structures may influence how genes are read, especially in the insulin gene, which we used as our model. This work was funded by the BBSRC and Diabetes UK. If this interested you, please do look at some of our other videos on the channel. Thank you for listening, and bye for now.